Hi, my name is Namita Ramesh, and today I will be talking about the effects of ITGAM genetic variation on Epstein-Barr virus or EBV transformed B cells. So what is systemic lupus erythematosus, or SLE? SLE is a chronic autoimmune disease that is caused by the overproduction of autoantibodies, which leads to the inflammation and tissue damage of various organs, like the kidneys, brains, lungs, skin, blood vessels, and even joints. However, the cause of SLE is still unknown. SLE is a chronic disease with an incidence in the United States of 3 to 10 per 100,000 people, and it currently affects an estimated 300,000 US citizens. The exact pathogenesis of SLE still remains unknown, but there is evidence that various genetic, hormonal, and environmental factors contribute to its occurrence and severity. Each of those is thought to modulate the activation and regulation of innate and adaptive immunity in ways that drive autoimmunity, which is the underlying cause of SLE. And central to autoimmunity in SLE is B cell hyperactivity, leading to the generation of autoantibodies. In a series of genome-wide association studies of SLE, among several single nucleotide polymorphisms, or SNPs, found to correlate with SLE were ones in ITGAM. The ITGAM gene encodes CD11B, which is one of the two chains comprising of the beta-2 integrin receptor MAC1, right here, which is CD11B and CD18 together. The ITGAM SNP RS1143-678 encodes a missense mutation that causes the amino acid substitution from proline to serine at position 1116 in the cytoplasmic tail domain of CD11. B. The cytoplasmic tail per se transduces signals important for white blood cells, which are the cells that express MAC1 to appropriately respond to pathogens. And serine at position 1142 in the CD11B cytoplasmic tail is known to be a site of phosphorylation, and the RS1143-678 encoded missense mutation introduces a second serine nearby that might also be phosphorylated and thereby render MAC1 on white blood cells from individuals carrying this SNP more responsive. So previous studies have shown that in SLE patients compared to controls, double negative 1B cells or DN1B cells, which are IgD negative, CD27 negative, and CXCR5 positive, are overrepresented. Then double negative 2B cells or DN2B cells, which is a subpopulation of DN1 cells, which are also CXCR5 negative and CD11C positive, are also overrepresented. So this study investigated whether SNP RS1143-678 altered CD11B/MAC1 function and how this might explain the association of ITGAM with SLE risk. And in order to study this, we investigated its impact on the biology of EBV transformed human B cells obtained from healthy donors. So by flow cytometry, we assessed expression of CXCR5, CD24, CD11C, IgD, CD11B, and CD27 to determine the relative distribution of DN1 and DN2 subpopulations. The diagram on the right shows the gating strategy we use to identify double negative cells, which are the DN1 and the DN2 cells, and also memory B cells. So degrees doublets and dead cells were sequentially gated out, which are represented by A, B and C respectively. Then CD24 negative, CD27 negative, IgD negative, double negative cells were identified by panel B, and the DN cells were further gated with the blue box in panel E representing CD27 negative, IgD negative, CXCR5 positive, the DN1 cell population, and the red box representing CXCR5 negative, CD11C positive, DN2 subpopulation. And finally, CD27 positive and CD24 positive cells, which are the memory B cells, were gated, which is shown in panels F and G right here. The increase in memory B cells led us to check for secreted immunoglobulins IgG and IgM in the supernatant of the B cell cultures using an ELISA test. So the results show a significant increase in CXCR5, CD24, IgD, CD11C, and CD11B receptor expression in EBV B cells. We found that the EBV B cells from donors expressing the RS1143-678 SNP variant showed also an increase in the DN1 subpopulation. CD11 or CD24 expression was increased in both the DN1 and DN2 subpopulations as shown right here. CD24 is a marker for memory B cells, and we found that there were more CD27 positive and CD24 positive memory B cells in culture from donors that carried the SNP variant allele. And using this as evidence of stimulation, we checked for secreted immunoglobulins, IgG and IgM, in the supernatant of the B cells using an ELISA test. So we found that 2 cultures of the SNP variant right here secreted IgM and IgG respectively, and 1 culture of the SNP invariant secreted IgM. Overall, both DN1 and DN2 cells populations showed an increase in the CD24 expression, which led us to test for both the memory B cells, which were CD24 positive and CD27 positive, and we also found an increase in the memory B cells for both the DN1 and DN2 population. And this data suggests that there could possibly be more immunoglobulins in the variant than the control. So since these cells were obtained from healthy donors, over time, these B cells could have spontaneously generated autoantibodies independent of the body. Then those B cells themselves could have generated the autoantibodies. So more experiments are needed to investigate the immunoglobulin secretion and autoantibody production in B cells from the donors who both have lupus and the ITGAM variation. But overall, an increase in memory B cells could lead to an increased autoantibody production, and further research on which kind of autoantibodies are produced could help us find the underlying cause of lupus. And I would like to acknowledge Dr. Tyler Wright for helping me out in this project. Thank you.